Chemotherapeutic resistance. When the patient is diagnosed as a cancer, and chemotherapy usually take the weeks of treatment. Unfortunately, some weeks later, the emergence of a resistance symptom in many cases appear. If that is the case, usually within a year, the full relapse is often observed. There are two types of resistance to chemotherapy. One type is intrinsic resistance. The tumor cells are heterogeneous. Some cells are intrinsically resistant to the chemotherapy. The other case is acquired resistance. Starting point of the tumor cells are responsive to chemotherapy, but when the drug compounds was added, intrinsic resistance cells they can survive, and some of those survived cells from acquired resistance they acquire new mutation for resistance. So after the treatment cycle is over, now those survived cells prevail and then relapse the cancer case. Drug efflux is one kind of a cancer resistance mechanism. Drug molecule enter the cancer cell, and it can bind to P-glycoprotein transporter, and by utilizing the ATP energy, and drug molecule is pumped out of the cell. And by replacing the ADP with the ATP, the transporter is ready to take another drug molecules. So in resistance cells, the P-glycoprotein is increased, and one of those representative gene is MDR1 gene. Here the MDR is multi-drug resistance. Another mechanism is a drug inactivation. Here the glutathione takes the important role. Glutathione contains a thiol group, and with the catalyst activity of GST, glutathione transferase, glutathione can be transferred to drug molecule and drug can be inactivated. And this complex molecule can be removed from the cell by MRP transporter. So in this type of the resistance, glutathione synthesis is increased. Also the enzyme GST and the transporter MRP is also increased. The third mechanism is a target alteration. Microtubule and topoisomerase, these are important cancer targets. Anti-microtubule or topoisomerase inhibitors, they can block the cell cycle and act as anti-cancer drug. Now the target microtubule and topoisomerase enzyme, they change their structure by mutation, and drug cannot bind to the target properly, and they escape from the chemotherapy effect. So this is the mechanism of target alteration. The last mechanism is quiescence. When the tumor cells are treated with the chemotherapy, majority of cells died, but some population, very small population of cells entered quiescence state, which is not actively growing state, then they can escape from the chemotherapy effect. After the therapy is ended, now these cells can return to the actively growing state and multiply, and then they can generate relapse of the cancers. Now if we summarize those acquired resistance and intrinsic resistance, drug efflux, drug inactivation, and target alteration, these are usually considered as acquired resistance, and entering quiescence is intrinsic resistance.